Okay, welcome back. Thanks for being on time. So we're done with that first exam, and I think that my first impression since I've marked the first page of most of them is that some of you have done rather well. And I hope that you might take a moment, even now, to sort of look again at that list of hints and think about how you did and how you studied. And I think what you would find that your memory of the exam would show that those were really spot on. And the big questions and the small questions related to them. So, what? Okay. I was waiting for that whine to continue. So, without regard to how you've done on this, and I'll give you some more advice when you get this back. I may have it done marked for you, and we may go over it in the lab tomorrow, or I'll give it to you on Wednesday. What other things do I want you to think about? Well, two things you have due tomorrow, or one thing's really due tomorrow, and that's text assignment number 5. The lab assignment you can hand in tomorrow for that bonus star, or you can hand it in on Wednesday, right? You have a quiz tomorrow on the diffusion lab, so read that and be ready for it. And next Tuesday will be your 15% first lab exam, okay? So that will include the lab you do tomorrow and the lab you'll do on Thursday as well. So it'll include everything from the first day up till then. Okay? Would you just give me a moment? I'm going to go see what that is because we're trying to tape these things and it's not going to work with that buzzing going on. Does anybody know what that is? A drill, thank you. Okay. Obviously, I wasn't successful in getting the guy to stop drilling, but he did look at me funny. So there you go. All right, that's the best we can do. All right. All right, so things moving along. In addition to the diffusion lab that you have tomorrow, I'm asking you to, today's lecture will come from chapter 4. Chapter 5, write this down clearly, skip it, okay? Skip chapter 5. There's like 2 little bits of information about photosynthesis that I want you to know, and that's it, so don't read it. So skip from chapter 4 to read ahead into chapter 6. All right? So let's take a moment to get back in sort of sync with the lecture material. This is one of those topics that will come up again and again. especially when we look at nutrition in a few weeks. We'll talk about energy and calories, and that will relate directly back to cellular respiration. We talk about breathing and the respiratory system. We'll review again about cellular respiration and the connections between those things. So it's important that you know this. There'll be much bigger questions about cellular respiration on your next midterm. So today you had a few points about it. The next ones you'll be expected to know real details about that. Okay. So I just want to quickly get back in the scheme thing. This is chapter 4 material. And we've covered this before. Let's swing through it briefly here. Order takes energy. It takes energy to create that order, and most importantly, it takes energy to maintain that order. That's what it is to be alive, is to have orderly trillions of cells. And as long as you can take in energy and process it and maintain that orderliness, you remain alive. ATP is the means that that will happen. In the biggest picture, plants do photosynthesis to make high energy compounds. Organisms like us rip those compounds apart to extract the sunlight energy, change it into ATP, and that's what we use. As I've talked about, the energy is in the electrons. That's what's energized in photosynthesis, and that's where your cells go looking for the energy, especially in the mitochondrial processes that we'll dive more deeply into today. The answer to the question on your exam, what's the name for the sum total of the chemical reactions? Metabolism. We broke it down into anabolic, building things, and catabolic, ripping them apart. Have a sense of what entropy is, and that matter is neither created nor destroyed. You're going to see that there's this very peculiar mechanism about how life works to extract energy. And it's going to work through this proton gradient thing. And so a reminder again, hydrogen is an atom that contains one proton and one electron. That's hydrogen. If you strip the electron away, you're left with an ion of hydrogen, which is exactly the same thing as a proton, because that's all that there is. Okay? So H+, you can call it a hydrogen ion, or you can call it a proton. It's the same thing. And that's what's going to be used to create a gradient. And in the chemical potential energy is in that gradient, just like the potential energy in water behind a dam. Enzymes are proteins. The shape of the protein that is an enzyme allows it to hold on to its substrate, and in holding it, allow a tiny amount of energy to transform that substrate into the products of that reaction. It holds the substance just so perfectly that the next input of energy is enough to change it, to break a bond, like in lactase transforming lactose into 2 monosaccharide molecules. It lowers the activation energy. Again, it's the shape fit of the substrate into the active site that allows that enzyme to catalyze the reaction, to make it happen with such ease that it can happen thousands of times a second for some reactions. That chemical reactions or metabolism is something that needs to be controlled. Enzymes work in series, in pathways, and sometimes they're needed and sometimes they're not. You don't have enough energy to do all the things you might need to do all the time, everywhere. So you conserve energy by shutting down pathways when they're not needed. And that's easy to see in this inhibition of enzymes. And again, it goes back to the shape of molecules, so that binding an inhibitor changes the shape of an active site, and in doing that, it stops a reaction from happening. And what you understand is that you stop one enzymatic reaction in a pathway, you stop everything from that point forward. Talked about this in the statin drug I took. By taking an enzyme inhibitor of this drug, I could stop this reaction in my liver. And it would stop everything beyond that, because everything beyond it depends on that working. And therefore, I would have less cholesterol in my system. It's an example of a drug that inhibits an enzyme. I was making videos this morning over in the library talking about vitamins, and specifically this vitamin, B2 and B3, the so-called coenzymes that are going to be electron carriers in the process I'll discuss in more detail in a minute. This molecule helps electrons get transported in this key energy production steps that we're going through. And in carrying those, it's absolutely crucial for you to stay alive and make enough energy to support the systems. So that's what B vitamins, this vitamin B2, you've heard of those. You know that vitamins are crucial to your health, and some of them are important in energy metabolism. That's an example of that. Okay. Take a minute with these things. So there's that riboflavin, B2. And I'm not sure exactly what this supplement thing is. But it's an interesting thing. How many of you take supplements like vitamins? I'm really interesting thinking about this. And I've, just like everybody else, I like vitamins. I used to take them. Okay? And you can see here that there's a daily value that you need of this. And that's a normal amount. And that this particular supplement has 882% of that daily value. And that's pretty standard as these vitamin supplements go. And here's the rationale that explains this. Humans think vitamins, and they think correctly, are natural. And they think, I really need vitamins. And that's true. And that the government comes out, the FDA or the Food and Drug Administration or the Canadian Health Advisory, comes out with numbers for these things. And pretty much everybody thinks, well, there's a little bit, I need that. But since these are so important, shouldn't I really take just a little bit extra? Or, gosh, what would happen if I took 8 times as much that I need every day. Certainly, I'll be healthy then. Perhaps I'll live forever. Never get old and wrinkly. Okay? And so, when you go down the list here, here's something that's available 5,000 times the dose that is the recommended minimum. It's kind of crazy when you think about it. You know, part of it even is, I'm sure, and I cop to this as well, it's like, what does the government know? You know? It's like, you know, this is like science, this is like vitamins. I should just take more. Everything will be okay. And it's really hard to hurt yourself with these kinds of vitamins, like 720% of that and 6,000 times that, you know? And the truth is, I probably mentioned this already, is that all these B vitamins are water-soluble hydrophilic substances. So that when you gulp down that horse pill of a B vitamin or that multivitamin, it's pretty hard to hurt yourself because you're just going to pee it away. Okay? And you know that if you ever deign to look down, because it's bright yellow, isn't it, when you have a little bit extra B vitamins in the system. But it doesn't really hurt you because you're just peeing it away. But the question that arises, why are you taking 5,000% if in the wisdom of your body it's saying, what the hell am I going to do with all this stuff? It's not like you can store it up for later. Okay? So that's worth knowing about. Now, in terms of vitamin C, a little more problematic, because although you just pee it away, and you only need 80 milligrams or something a day, not whatever, okay, it acidifies your urine and it damages your urinary tract. So if you think this is completely harmless, you're wrong. Now, we'll talk about vitamin A and E, and K and stuff, and these are different. D and K and E and A are fat-soluble vitamins. And if you want to go home and Google hypervitaminosis, it's a really bad thing you get when you eat too many fat-soluble vitamins, because those you don't pee away. And those stay in your body, and they can cause liver toxicity and other problems. You can turn orange at a minimum. Okay? So vitamins are not neutral. You can pee a lot of them away, and and other kinds are just plain damaging. Okay? Now, this next part. This zinc, selenium, copper, manganese, molybdenum, chromium, all those things. What do you need those minerals for? And I mentioned this last time, and I mention it again. You need these as enzyme cofactors. Different enzymes in your body, some of them require a mineral ion to be connected to them for them to work. And I mentioned one last week, it was DNA polymerase, and it needed zinc to work. You need copper in your electron transport system for it to work. So if you ever wonder about vitamins and why they are and what they're doing, now you have a sense. Some are coenzymes and electron carriers. Some of them help enzymes work. But all of them are involved in these basic functions of enzymes and the electron transport system. We'll get back to this point about essential amino acids when we talk about diet in the next weeks. Okay. Here was the ATP that was on your exam. Yes. Yes. So let me explain. So, used to do experiments, and you need to extract things in different kinds of solvents, and like DNA and stuff, you could extract in watery solvents, okay? And other things you need to extract in non-polar solvents, things like benzene and stuff. And so what it means is that if you have a fat that's non-polar, and if you add a non-polar substance to that, that all mixes together. It's quite happy. Yeah. Oh, how interesting. There are hydrophobic attractions, but it doesn't work in opposite. So he's absolutely right. So think about this. When you put something in water and it dissolves, it's because you got like sodium is positive and chloride is negative, and the different ends of the water molecule can surround either one and bring it into solution. For something to be non-polar is to have the absence of charge. And the absence of charge will merge really well with something else that has an absence of charge. So if you were to mix like olive oil and sunflower oil, you could mix those together perfectly. They wouldn't have any repulsion. They're not attracted to each other in that sense, but they are not adverse to each other. Okay? And so to finish that story, when we'd use this nonpolar substance, it was like a horse liniment thing. And if you spilled some on your finger, if you weren't wearing gloves, then in just about a few seconds, you'd get this really weird metallic taste in your mouth. And that was because, essentially, this is going right through your skin, right through your cells, right through your body, and you would be able to even taste it in just a few moments, because essentially it can just flow through all your cells. Right? So that's that matching of nonpolar and nonpolar. Here was the answer to that question in your exam a minute ago. 3 parts of ATP, adenosine, sugar, and 3 phosphate groups. Okay, so I already previewed this for you. We're ripping apart molecules to get their electron energy, and the captured electrons that we rip apart mainly in the citric acid cycle are going to be carried by the coenzymes, the B vitamins, NAD and FAD. And when they gain electrons, they'll be reduced, and when they give up those electrons, they're said to be oxidized. So you're going to gain electrons, reduce, lose electrons, oxidized. And so what these carriers are going to do is literally act like, do you ever watch the harbor ferries downtown? Okay? They're these little boats, and they're green, and they do that little ballet downtown, if you've ever seen that. And so people get on those, they go for a little ways, and they get off these things. So if people were electrons, when they stepped onto the ferry, they would reduce the ferry, and when they step off it, they would oxidize it. And these things act in that way. The electrons that you rip away, they step onto an NAD. The NAD goes to the electron transport system where they step off, okay? And now it's oxidized. And now that boat goes back to pick up more and returns with them. So it's constant being loaded and unloaded in uncountable numbers of these things to deliver these electrons to create the proton gradients to capture the energy. Okay? Now, last time I gave you very clear instructions about what you need to know about these 3 parts, and these are the things I can ask you on the next exam. What happens? Where does it happen? What energy-rich molecules are made? What goes in? What comes out? So, glycolysis, I think this is just where we left off last time. So glycolysis happens in the cytoplasm. The input is just straight up glucose. And we're just looking at this for now. The only thing we'll look at that's different a little later on is to say, well, how do fats flow into this process and how do amino acids? Because you can get energy from protein and you know you can get energy from fat. And they're going to use these same systems virtually identically. So we're just talking about glucose, but you know when we're talking about this, this is how you're going to extract the energy from fats and proteins as well. Okay? Put them in, you take that out. Anaerobic means that this happens whether that cell has oxygen present or not. You charge it, you sort of supercharge it with a little bit of ATP input, and you get a little bit net out. But the reduced coenzymes, and write this down, are really the energy-rich molecules that you care about because it's from there that you will get the vast majority of energy output. And we're doing this just to follow the carbon. So you put in 6 in a glucose, you charge it up. Just to be clear, those little PIs, that little subscript I, don't be concerned about that. It stands for inorganic, and you put it on there all the time. And it just means that those phosphates came from those ATPs. And when you take off those 2, you're left with 2 ADPs, adenosine diphosphate, so tri to diphosphate. Everybody clear on that? Then in a bunch of steps, you run these through to make pyruvate. End of glycolysis. These end products form the input for the next stages of the process. And there's 2 possibilities here. Let me say it and then we'll see it again. If there is no oxygen around, you ferment those pyruvates. Fermentation is something that happens in the absence of oxygen. And if you do that, you make lactic acid. End of story. You cannot live if you don't have oxygen, and the reason is that you don't make enough energy out of just fermenting pyruvate to live. You've got to have oxygen around. So this will only occur under those conditions like in muscular exercise where you've exhausted the oxygen supply and you haven't replaced it yet. So it really hurts, and so you're supposed to stop and let the oxygen regather there to continue on a productive path. Okay. So here in this next part, and this first couple steps I called the grooming reaction. Here's one of those 2 pyruvic acids. We rip some electrons away to reduce this coenzyme. You split off a carbon in the form of carbon dioxide. Every single carbon that was in the glucose, all 6 of them, eventually you're just going to breathe out. That's what happens to those. But before they're breathed out, you take away what you wanted, and that was the high energy electrons. So there's some more reduced coenzymes. There's what's left over, the so-called acetyl-CoA, and that's the input for now the 3rd part of the process, called the citric acid cycle or the Krebs cycle. The input, acetyl-CoA. You'd get 2 of those per glucose. Where does it happen? In the matrix of the mitochondria. What are the outputs? Well, you get reduced coenzymes, 3 and 1. So you'd get 6 and 2 if you were counting in terms of each glucose, because this would spin twice. And the rest of the carbon atoms, 2 there, get expelled as CO2. You make a little bit of ATP directly, but it's from these molecules that the vast majority of the energy will be derived. So let's step back and think about a concept you learned earlier about isotopes. So it really wouldn't be unethical of me, if you approved, I could feed you some sugar that was made with carbon-14 instead of carbon-12. Or I could feed myself. And if I had the right kind of equipment, I could capture the air that I breathe out, and I could test that in a mass spectrometer. And I could literally answer the question. So if I drank a glass of orange juice that had carbon-14 sugars in it, I could see how long it took me to take those in, to digest those, take them to my liver, send them to my cells, burn those in the mitochondria, and breathe out that carbon-14. And I'd be able to track that and see, if I was interested in, how long that took from ingestion to breathing out those molecules. So that's where the carbon dioxide, the carbons in the sugar that you eat, end up getting breathed out as carbon dioxide. So here's that grooming reaction that I talked about, and then here's the citric acid basics. Now, it says that this part absolutely requires oxygen to work. And it's interesting because you didn't see any oxygen anywhere. And really the truth is that it's the next step, the electron transport system, where oxygen is actually used, but that the whole system will clog and back up if oxygen isn't present. And so this will stop because there's no place for these reduced coenzymes to go drop the materials off. So if you clog up the last part, this part can't go either. Okay. And so here we are at the very last step. So we've gone through the Krebs cycle, in the matrix of the mitochondria, did the grooming, we did the glycolysis before it, and now here's the big energy payoff. So pay attention. Here's this reduced coenzyme. It's carrying electrons from glucose. When it drops those off, that's what that arrow represents, is the flow of electrons. See there, 2 electrons? That's what this NAD dropped off, NADH. And then it was passed along, those electrons, and eventually it combined with oxygen and protons and made a water molecule. So the oxygen that was sitting there is now gone. It can't do its job anymore. It was there to pull those electrons through. It did that. It got those electrons. It's now part of a water molecule, and you need another oxygen there. So imagine that if that isn't there, then those electrons don't have anywhere to go. They get stopped there and there and there and there. And now you can't drop off anymore. And that's how come the Krebs cycle would stop. That make sense? Notice this. At each of these mitochondrial proteins that you see here, when the electrons get passed, the protons get moved out into this intermembrane space. And they're stuck there. It is literally like the solid wall of a dam. Those protons, once pushed there, can't get out. And they're building up and they're building up. And gradients, water or protons, represent energy. and that here is the only way for those protons to go in the direction that the universe wants them to go, in the direction of making things even again, allowing entropy to happen. Okay? You got them really highly organized. To relieve that organization, pass them through this molecule. It's called ATP synthase. And it shoots them through like a water slide and then collects the energy in the bond of ATP here. So for every pair of electrons that NADH drops off, 3 protons are pushed into that space. For every electrons that FAD pushes in, they only push out 2. Write this down. Every proton in that gradient makes an ATP. Every proton in that gradient makes an ATP. It's often really instructive to understand how things work by how they fail or how they can be poisoned. This system depends on this membrane being absolutely impermeable to protons. In the 1930s, they discovered what seemed like a wonder drug at the time. It was called dinitrophenol. And if you took dinitrophenol, you got some pretty remarkable weight loss things happening. Now, sure, a bunch of people who would take it would die, but, they died looking well and skinny. And the way that this dinitrophenol worked, and they didn't know this at the time, but they do now, and if you Google this, you can still find people who will sell this to you. But here's how this poison works. Dinitrophenol is nonpolar, hydrophilic. It comes into your cells and goes into your mitochondria and goes into your inner mitochondrial membrane. And it makes that membrane leaky for protons. And so your cells continue to burn food, and in the expectation that they're going to get the payoff of the ATP from burning that food. That's the point of doing it. But when you're poisoned with dinitrophenol, you don't make any ATP because you don't make a proton gradient because the membrane is leaky for protons. So, can you take a moment with your neighbor and describe the death of people who would be poisoned with dinitrophenol? Do this already? Talk about this? Can you think about it? So instead of making ATP, how would a person die if they poisoned themselves with dinitrophenol? Go ahead and think about it. I mean, the answer is just a couple of pages away in your notes. But the point is, is that you would die from overheating. You would cook yourself. Okay? So here's the grand scheme of energy production. You take glucose in the presence of oxygen and you burn it really slowly. You make carbon dioxide and water and energy. About 35-37% of that gets captured as ATP. The rest gets made as heat. Gets released as heat. If you don't make a proton gradient, then everything you burn turns into heat energy. And since your systems are struggling because you don't have the ATP, you burn more and more of the food energy, creating more and more heat, and therefore you get warmer and warmer and warmer. And if you didn't pay attention, it would just be like a catastrophic fever. Your temperature would spike dramatically, it would go over a point, and then your proteins would denature and you'd be dead. Okay? So if you go and look at these sites, the people who still sell it to you have some semblance of conscience, and they do warn you to be careful about overheating yourself. What else is interesting about this is the different kinds of fats that humans have. You have mostly at this stage of your life stuff called white fat. But when you were a little baby, you had brown fat. Babies are really temperature sensitive. And to help them just generate heat, they have more brown fat cells. And what brown fat cells is the same as dinitrophenol does. They are cells that are just there to burn fat, but just to turn it into heat to help this baby stay warm. And so, yes, scientists are working really hard to try and program a little proportion of adult white fat cells back into brown fat cells, because just like DNP, if you had just a twitch more brown fat, then essentially you could burn 200 or 300 extra calories a day, and hence have that sort of idealized state as being able to eat whatever the hell you wanted and still just burning it off. Okay? So here's the stuff that I've talked about already. The reason that oxygen's there is to pull the flow of electrons through. As the electrons flow, the protons are trapped in that space. The proton gradient has energy. ATP synthase captures that energy by a process called chemiosmosis. The inputs for the electron transport system are those reduced coenzymes, those B vitamins I talked about. And the outputs are water and about 36 ATP per glucose. Here it says 34% efficient in capturing energy as ATP, so the vast majority as heat. And the place that this happens is in the inner mitochondrial membrane. So on your next exam, I can ask you, describe glycolysis, or Krebs cycle, or citric acid cycle, and the electron transport system. How does the electron transport system work? And you'd have to be able to sort of describe or draw pictures about how this energy transformation occurs. You take a moment with your neighbor and see if there's anything you'd like to ask now to make it clearer. Okay. Is there anything I can help with? So here it is, talking again. I talked about DNP. I didn't talk about cyanide, but that's an interesting example as well, too. If we flip back through here. Cyanide, I believe, comes right in between this cytochrome C and this other electron, part of the electron transport system. And it bungs up the system. And what happens is that you can't make the electrons flow anymore. Blocks the flow of electrons, just like the absence of oxygen does. And so, just as if you were suffocating without oxygen, the presence of enough cyanide in there would block the flow of electrons, prevent the formation of ATP, and literally you've run out of the energy to respire. You can't raise your chest wall, you can't contract your diaphragm, you can't breathe anymore. You don't have the ATP to continue having your heart beat. So you just run out of energy and you die. I mentioned this before. Anaerobic parts of things. So glycolysis is anaerobic, and what happens when you don't have oxygen. So we talked about lactic acid caused by the lack of oxygen in muscle cells, okay? And it's sending you the very powerful signal to like calm down, sit down, take a break, in order that the oxygen can be restored so that you can start making adequate amounts of ATP. You only make 2 ATP out of essentially the 40 that you make in total. 2 of them is made in the glycolysis process. That's not enough of an energy return to keep you alive. 2 out of 40. Now, some organisms can kind of calm down and live with that minimal energy input. Things like yeast can survive with oxygen and without oxygen. You cannot. And so here's lactic acid fermentation. This would happen in your muscles in the absence of oxygen. And this is what would happen in yeast. So if you're making beer or wine, you'd add the yeast, have the maltose sugars or the grape sugars, you'd put a lid on it, and you'd put a little stopper cork that would allow the carbon dioxide that's bubbling away in order that that would exit, otherwise your vessel is going to explode. And it would also produce ethanol. So it would chew through those products of glycolysis, and then in the absence of oxygen, because you've got the lid on it, it would produce carbon dioxide and ethanol. This process, invented by bacteria, we talked about how your mitochondria are remnants from 1,200,000,000 years ago that became part of what we call eukaryotic cells. And even though you are only very distantly related to yeast, you do this process just like they do. Exactly the same process, but for the fact that they make ethanol and carbon dioxide when they ferment. If oxygen is present for yeast and oxygen is present for you, they do it exactly the same way. In cell cultures, you can even get in there and move stuff around. And so in cell cultures, you could take electron transport systems from yeast and put them into human cells, and they'll work fine. And you could put yours into yeast, and they work fine. Okay? Once a system like this is invented evolutionarily, it doesn't change very much. People were massively surprised when they discovered the fruit fly developmental gene pathways. Nobody expected that humans would have the same genes acting in the same order to key the developmental stages of humans. In fact, what they used to find the genes for developmental processes in humans were using fruit fly genes to go hunting for them. Because although they're very different organisms, those kinds of processes are extremely conserved in nature. So likewise, the way in which we make energy is largely the same in all eukaryotic organisms. Now, as I promised, we talked just about what happens to glucose as it goes through glycolysis, grooming, citric acid cycle, electron transport system. There isn't a whole new set of things when you derive energy from other food molecules. So, carbohydrates and some amino acids, a piece of some fats called glycerol, can all be processed in the same way that glucose was. Okay? Now, when it comes to the long fatty acid tails of lipids like triglycerides and things like that, they don't go through glycolysis. They get made into pyruvate and shoved into the Krebs cycle. And some amino acids can actually just enter the Krebs cycle directly. But all of them undergo the same kind of transformations. What you are doing is ripping apart the fatty acid tails into little chunks of 2 or 3 carbons. And just like in glucose, those carbons are going to be breathed out as carbon dioxide, the high energy electrons gathered to make ATP. Okay? So the energy gathering portions of these reactions use the same systems, just slightly modified to extract the energy from other things. When we do our diet diary lab in a little while, we'll talk and we'll look at food labels, and you'll learn that there's more calories per gram in fats than there are in carbohydrates and proteins. And why that is simply the source of energy is always those high energy electrons. So per unit weight, there are more high energy electrons in fats than there are in sugars or amino acids. So that's why there's more calories there. And in evolutionary terms, that's how come evolution has trained you to want and crave fats. In our evolutionary history, calories were scarce. If you had to choose between things, you wanted either high sugars like ripe fruits, or if you're eating animal stuff, you want high fat material, plant oils and animal fat, because those would give you the most energy per bite. Okay. So take another minute with your notes here, and depending on what you say on Wednesday, I can walk through these portions again, but I wish you to take a whole minute with your neighbor now to flip through these parts and see if there's other things you want to ask, and then we're going to go on to chapter 6, depending on how many questions we get. Okay, go. Okay, so you've had a chance to look over those things. Is there anything that I can help you with? Okay, well, I encourage you, since this will be a significant chunk of the next exam, if you decide on Wednesday, if you want to review portions of these, I'll be more than happy to go through them again. It often takes a few times through. May I suggest that you look at those Mr. Anderson videos or anything else about these topics, because you have it in the book, you have it from me, you have it on PowerPoints. But another viewpoint can often help really anchor this in your mind. And do please remember those instructions about how much you need to know about these things. It is limited. I don't expect that you know every stage. I don't expect you to remember every intermediate in the electron transport system or in the citric acid cycle. Okay? Now, there was a portion of chapter 5, and I wanted to make sure that you looked at this, and I may have even shown you this before. Here's the equation for photosynthesis. Would you take a minute and write that down? I'm pretty sure I've already shown this to you in the context of talking about chloroplasts and mitochondria. Yes, I'm sure I have. So carbon dioxide, the very stuff that you are breathing out, is the gas molecules that plants take in through their leaves, along with water from their roots. And in those organelles, the chloroplasts, those get combined to make what you know here, the chemical formula for glucose. They make sugars. So they take something with no energy that is available and make something that's a ready energy source. Energy comes from sunlight. Energy is put into the electrons that make the bonds of glucose. And meanwhile, just by the way, they make the other thing that you require for cellular respiration to go, the oxygen to draw the electrons through the electron transport system to create the proton gradient to make energy. And you know that if you simply change the arrow of the direction of this arrow, you take in sugars in the presence of oxygen, make water, carbon dioxide, and ATP energy and heat. So just turn the arrow around and you have what's happening in your cells. Just be clear so that you don't make any silly mistake. This is what plants do. Cellular respiration is what you do. But please understand, plants do cellular respiration as well. Plants have chloroplasts and mitochondria. So they just make their own food. They're called autotrophic organisms. But they make those sugars, etc., and then they burn those sugars in their cells just as you do. Okay? You are called a heterotroph. Means you eat other things. You don't make your own food. Plants have photosynthesis and cellular respiration, and you just have the one cellular respiration. So here's a big sort of inflection point in the course. We're leaving behind the first third of the course and going on to the next part, which will have topics about cell reproduction, both mitosis and meiosis, and then we'll go into a section on the basics of genetics and inheritance. We'll talk a little bit about the genetic code, tiny bit about cloning, genetics of cancer, genetically modified organisms we'll touch on as well. Okay? So you can draw a great big double line through your notes. We're moving on to chapter 6. So let's begin. You're made up of 37 trillion cells, or at least I am. No, I'm probably more because I'm bigger than average. If you're average-sized human male, 37.2 trillion cells. With some uninteresting exceptions, every one of those 37 trillion cells has a nucleus, and in that nucleus, in humans, there are 46 chromosomes, and those 46 chromosomes are fundamentally identical to the ones that you got from your parents, and you got 23 from your mom and 23 from your dad to make a total of 46. When we talk about cellular reproduction, we're talking about, in eukaryotic cells, the process is called mitosis. Write it down. Mitosis is a process of nuclear division. Actual splitting apart of cells is a different thing. It's called cytokinesis, and we'll look at it in a minute. But that's the actual division of the cells. Mitosis technically refers to the division of the nucleus. The process of mitosis is called equational. Start out with 46 chromosomes in one cell, and you end up with 46 chromosomes in 2 cells. The cells are called daughter cells for some reason. They're genetically identical to each other. This is the way that you grew from a single fertilized egg into the 37 trillion cells. All your cells, barring the mutation, are genetically identical to each other. So there is no genetic difference between your retina cells in your eyes and your liver cells and your kidney cells. It's all exactly the same information. The tissues are different, write this down, because they express different sets of genes. But all of them have all the genes. They just express different sets of genes, like the liver set, or the kidney set, or the retina set. But they haven't gotten rid of anything. If you're going from 46 chromosomes in one cell to 46 chromosomes in 2 cells, you got a copy. And a critical portion that happens before mitosis is that all the DNA, write this down, 3.2 billion base pairs of DNA has to be copied. And then those 2 copies have to be perfectly separated. So that the daughter cells are identical. And I mentioned this a couple times already. The key enzyme in this complicated process is DNA polymerase. DNA polymerase can work on each half of the DNA molecule, essentially taking one strand, and because of the very simple base pair rules that you know, A pairs with T and G pairs with C, having one strand is all the information you need to make its complementary strand, and likewise for the other strand. So you can start out with one double-stranded molecule and end up with 2 double-stranded molecules that are identical to each other. And therefore all the information that is in the sequence of those bases gets passed on to the daughter cells. Make sense? So in this first picture, you could see here, you would know that this is an animal cell, because you've seen plant cells in the lab. They have square boxes around them. This is an animal cell. It's nice and round. And here are little relations of muscle proteins that are coming together to kind of twist those fat bubbles apart in the creation of two new daughter cells. Inside each of these cells already would be the whole complement of sea urchin DNA. Okay? And so as these divide and are held together, each one of those new cells would have the whole suite of genetic information. Lots of terms here. Let's start. First term you run across is karyotype. For most people, if you ever run across karyotypes, it would be usually of an unborn child. A karyotype is just a display of all the chromosomes in an organized way. You have 46 chromosomes, you got a karyotype done, or you had a karyotype done on an offspring. It would show those in order. Oh, and chromosome numbers, like what's chromosome number 1 and 21 and 22, how are they organized? Just by size. The biggest chromosomes are chromosome 1, and the smallest ones in you are chromosome 22. You have a pair of sex chromosomes too. So a karyotype is just a display of the chromosome pairs from small to large. You, and really most of the things that you encounter in your life, maybe except yeast and mushrooms, are called diploid organisms. All that means is you got 2 copies of everything. 1 copy from your mom, 1 from your dad, 23 chromosomes from mom, 23 from dad. You got 2 copies of everything. So what this means is, in practical terms, is that in a group of 40, there's likely 2 of you are carriers of cystic fibrosis. Okay? What it means to be carriers is that you have 1 good copy of the gene and 1 mutant copy of the gene. You're perfectly normal because you're diploid, because the 1 good copy is enough. Right? You'd have to have 2 mutant copies for you to have the disorder. So diploid just means you have 2 copies of everything. Now later on, we'll look at the process in your gonads, testes and ovaries, by which you make sperm and egg, and that process would make haploid cells, which just means that you've taken 1 of each of the pairs picked randomly and put those into a sperm or put those into an egg. So in you, 46 is the diploid number, 23 is the haploid number. Half the genetic information, 1 copy instead of 2. Here is a, let me emphasize this, false color image of a human karyotype. Your chromosomes don't glow different colors. They have cool new techniques that allow you to paint chromosomes to identify specific ones in order to do interesting and useful, not just cute things. Okay? So here are the 22 pairs. That makes up 44 of the 46. And you probably know that you have a pair of sex chromosomes as well. If you're female, as is shown in this karyotype, you have 2 identical sex chromosomes. They're X's, they're great big ones. If you're a male, got a great big X and a little stubby Y. Okay? That Y determines maleness. Okay? So chromosomes don't come in these orderly sets. Chromosomes get splayed out on a microscope slide like this, and then you use a little Mac program to kind of lasso them and put them over in the right spot, and that's the way that you create that. Now, why would you do this? Well, karyotypes of cancer cells are often bizarre and interesting and help in diagnosis and treatment. But most often this is done on unborn childs of mothers over 36 years of age, where they're looking for chromosomal abnormalities. The one that everybody would know, of course, would be Down syndrome, where instead of having 46 chromosomes, you'd have 47, and the extra one would be an extra 21. The reason you don't know the names of 22 other chromosome abnormalities is because they're nearly always fatal. You can think about it as Down syndrome is the least bad thing that can happen when you get the wrong chromosome number put into a gamete and hence into the next generation. Do you have questions about this karyotype that I've just put up here? Yes. There are peculiar things that can happen, and you can get so-called chimeric individuals and things like that. But basically, no. There's no fixing that. And remember the scale of the problem. You got 37 trillion cells. There's no surgery you can do to. I understand what you mean. And so what would happen there is that you could, and in certain cases people do, who are known carriers of really bad genetic disorders, they could test their embryos not for extra chromosomes, well, they could do that as well, but you could do it for a genetic disorder. And you wouldn't necessarily go in and fix that, but if you were doing this in vitro thing, you might have a whole set of fertilized eggs to work from. You could test them and pick the ones that didn't carry that disorder. So, these kinds of mistakes we'll get to. So she's asked, so these kind of mistakes in number that we talked about are going to be mistakes in meiosis. They'd happen in your testes and ovaries, okay? And they would potentially go into the next generation, and what happens sometimes is like Down syndrome would be the result, okay? And so, but I think at a deeper level, your DNA doesn't have any sense of what it's supposed to be. Your DNA polymerase is going to copy DNA, whatever task it's given. It doesn't sort of judge it, or, well, it can fix some things in some ways. It's complicated. But basically, it's just a machine to copy things. If it had an extra chromosome there, it's not going to know there's something wrong and kind of fix it. Helpful, yes? But that's where it went wrong, and it goes wrong a lot. So if you ever want to really understand what a fallible, evolved creature you are, is that everybody probably knows somebody who's had a miscarriage, and a huge percentage of miscarriages are caused by the fact that people make sperm or eggs with the wrong chromosome number, and nearly every time you do that, it's fatal to the developing embryo. So it happens a lot, and it's just a mistake in the processing of chromosomes in a kind of complicated dance of these chromosomes separating from each other. It doesn't happen perfectly. Just make lots of mistakes that way. And it turns out, for both males and females, as you get older, you make more and more of those. That's how come, you know, if a woman was 20 and pregnant, they wouldn't offer her this process of amniocentesis and karyotype analysis. And if she asked for it, they'd say, in the absence of any evidence, we can do this, but you're going to pay for it. If a woman at 39 went in, they would say, would you like to get this done? You are at greater risk. And she can say, no thanks. Or she can say, sure, I'd like to know. And they would pay for it because you're now at a greater risk for chromosomal abnormalities. Anything else? Now here's yet another term, and there will be many more of these. So you saw back there were 23 pairs, and each one of those pairs, the two chromosomes, are called homologs, homologous chromosomes. And what my grandmother would say is that they're exactly the same, only different. They are exactly the same in that they have exactly the same genes in exactly the same locus or loci, and that just means like the address of the gene. You might have 1,500 genes on chromosome number 1. And since you got 2 copies of that, you got 1,500 genes on each of those. And each of those genes has a chromosomal address, and it's always the same address. But the versions of those genes can be different. They could have the eye color gene there. They could have a brown eye. They could have an eye color gene, and we'll just say it's brown or blue. And they could both be blue, they could both be brown, or one could be brown and one could be blue. Okay? So they can be exactly the same in the genes that they have, but difference in the versions of genes that they have. Does that make sense? Now write down this part. The version of the gene versions are called alleles. Alleles of a gene. And although eye color is more complicated than I let on, let's take a look at it. You have, write this down, about 23,000 genes. And therefore, your diploid body cells have 46,000 alleles. One for each. I probably should have said something like 21,000 genes and then be different than the chromosome number, which is kind of confusing there. I can't remember. So let's say 22,000 genes instead of 23. I think that might even be closer. Right? And therefore you have 44,000 alleles. We'll just take it from that 23 and 46. So if you think about a chromosome, they're often written like this, and here's a place, and we'll say, here's a dominant large B brown eye allele. And maybe you inherited that from your dad. Have another homolog, and at exactly the same location, and this one you got from your mom, because you got one from your dad and one from your mom, you might have the recessive blue eye allele. So these are homologous chromosomes, and this is one gene, the eye color gene, but you have 2 copies of it, and one version is a dominant brown and one is a recessive blue. And we'll cover this again and again. But I want to emphasize this idea of the 2 copies of each gene, 2 alleles for each gene, and they can be the same or they can be different from each other. So everything that's eukaryotic, plants and animals and fungi and protists, all use cell division. And that cell division is always called mitosis. And the function is to make identical daughter cells. And as I've talked about, all your cells are genetically the same. So what do you use this for? Well, you used it to go from a single egg, fertilized egg, into eventually an adult human. You get damaged. You can repair some things. You can get cut and fix things. That would be repaired, make some new cells. You're constantly shedding epithelial cells like we talked about in the lab. If you got stabbed in the liver, your liver could regrow some portions of it. Doesn't normally do that. But you could repair things. You could make things, you could fix things. Oh, God. Okay. So, what a perfect place to stop. Thank you for zipping that up. It reminded me. And so, we'll see you tomorrow morning at 9:30 in the lab. Bring those things that are due. I will try and get your exams done, and we'll see you tomorrow.